Okay, this is the second half of chapter 22 where we were talking about speciation, and we just went through the process of allopatric speciation, which is speciation that results from geographic separation over a long period of time, physical separation. Now, sympatric speciation, which is the other main model for how speciation could occur, is speciation without the physical isolation. And sim always means together, right? So here we have organisms that are still living together, interacting with each other, but we're still getting speciation. So this is a rare event, but I'm going to give you two examples of how this could occur. And really, one thing to keep in mind is that even though organisms may be living together, at least from our perspective, in the same physical area, keep in mind organisms can become specialized for very specific, what I'm going to call microhabitats, right? So, very specific food sources or very specific places where mating might take place within the same general physical area. Um, so an example of this is sympatric speciation that seems to be happening right now in this organism pictured here, which is the apple maggot fly, which we find primarily in eastern North America. And so this fly had been around before European immigrants brought in apple trees. And even though these are now called apple maggot flies, originally these flies basically only deposited their eggs on the fruits of hawthorn trees. So they would lay their eggs on these fruits, right? That would provide a nice sugar source for development of the maggots into flies. But along comes Johnny Appleseed, and European immigrants start planting apples everywhere to be able to produce hard cider. Is originally why they were planting the apples. And these flies realized, oh, well, the apples, make a nice substitute for the hawthorn fruit. So some of them began laying their eggs on the apples instead. So these are sort of two different microhabitats within the same general area, right? You may have an area where there's hawthorn trees and apple trees, in close proximity to each other. But we think we're in the midst of some reproductive isolation here because we see, if we look at the populations of these flies that are laying their eggs on hawthorn versus apples, they tend to mate primarily with individuals that were raised on the same fruit. And part of this, at least, is due to just the timing. Um, so, because hawthorn fruits and apples are going to mature at slightly different times, you end up with the maggots from these flies developing at slightly different times. And because flies have a very short lifespan and a very narrow window for reproduction, just the timing of the fruiting of those trees is causing some reproductive isolation between these different populations. So we're going to have to see how this plays out, but we may reach the point where we actually have two distinct species of flies, but it still seems to be in process. Another relatively common way that you could get sympatric speciation is really something that only typically happens in plants, which is this phenomenon called polyploidy. And I don't know if you remember back to the mitosis chapter, we talked about aneuploidy, where you have an extra copy of or a missing copy of one chromosome. So it'd be like Down syndrome, trisomy 21, right, would be an aneuploidy. Well, polyploidy is where you have abnormal numbers of all the chromosomes. So instead of having two sets of all the chromosomes, you've got three sets or four sets. Um, and animals do not tolerate polyploidy well. There are a few examples of polyploid animals, but plants, because they're a little bit more flexible in terms of their developmental process, seem to tolerate this polyploidy a lot better. And in fact, a lot of the common like crop plants that you know have very high ploidy numbers, 8, 9, 10 sets of chromosomes. Um, but what can happen is, so you can have polyploidy basically just accidentally happen if two diploid gametes manage to successfully combine with each other. So this basically would be an error in meiosis, right? So imagine you have a diploid plant. It should be producing haploid uh, pollen or eggs, right? Um, but due to accidents in meiosis, you end up with diploid sperm, diploid egg. If they combine, you now have a tetraploid plant, so 4 copies of every chromosome. So you've got this unique tetraploid plant, right, surrounded by all these other diploid plants. And if that tetraploid tries to breed with the surrounding diploid plants, um, you're basically going to have gametes that have 2 copies of every chromosome from the tetraploid and 1 copy from the diploid. So we bring those together and you're going to have 3 copies, triploid. And anytime you have odd numbers of chromosomes, those organisms are always going to be sterile. So you've essentially got a evolutionary dead end there. But keep in mind, a lot of plants can actually self-fertilize, right? So that tetraploid plant is now reproductively isolated, right, from all the diploids around it, but it can start reproducing and repopulating with itself. So it can rebuild this new tetraploid population that is now reproductively isolated, and by the biological species concept, would be considered a new species at that point. So this is something that can happen really quickly in just a couple of generations in plants. So polyploidy driving sympatric speciation. All right, so to finish things up here, I want to talk about a couple ideas. One was I wanted to go through some of the barriers that can help enhance this process of speciation, and then we'll talk just really quickly about hybridization and some of the genetic traits that may affect the speed at which speciation occurs. So we've talked about reproductive isolation, and researchers will categorize reproductive things that can drive reproductive isolation into these 2 categories. So what they call prezygotic barriers or postzygotic reproductive barriers. And remember, the zygote is the fertilized egg. So a prezygotic reproductive barrier would be something that's going to even prevent fertilization from happening, right? Prevent the zygote from being formed. Whereas a postzygotic barrier would be something that happens after fertilization, but that is still basically preventing the future development of this population into a distinct species. So okay, so let's look at some of these examples. So these are all what we would call prezygotic barriers. And we've got habitat isolation, it's called temporal isolation, behavioral, mechanical. And I'll just real quick kind of give you an example of each of these. So habitat isolation would be, let's say you have, 2 different populations of snakes. One's a water snake, one lives on land. So these 2 populations might be physically capable of interbreeding with each other and producing offspring, but because they are spending their time in these very different habitats, right, that is going to encourage that reproductive isolation and help drive the difference between those 2 populations over time. Temporal always refers to time. So this could be a situation where you've got, 2 populations where one is nocturnal, for say, and one is not, or might even refer to, time of the year where they prefer to interbreed, something like that. But if we have these significant differences in behavior related to time in some way, that could also help drive differences between populations. Behavioral isolation We see this in particular with birds, where the females of a species are usually looking for some very specific either appearance or in this case behavior in order to choose a mate. So these are male blue-footed boobies. You find these in the Galapagos. They've got these bright blue feet, and they have to do this very specific dance. You can see the guy on the left here is, doing his dance and flashing his feet around. And the female is going to watch the dances and decide, who did the best job or who she thinks did the best at displaying their blue feet and choose a mate that way. So you've got this very specific courtship ritual. So, there may be some other population or species of bird that physically could interact and mate, but because it doesn't have the right behavior, right, the females are never going to want to mate. So behavior can drive reproductive isolation. And then you have mechanical isolation. This goes back to the example I was talking about with the snails. So this would be a square peg in a round hole kind of situation where parts just physically do not fit together and they can't interbreed. Remember I was talking about the fact that one gene can control the direction of the spiral on the side of a snail shell. If you have opposite spirals like you're seeing here, parts don't line up correctly and can't interbreed, mechanical isolation. The last prezygotic barrier is what's called gametic isolation. And this means that the gametes, the sperm and eggs from two different populations or two different species, just biochemically cannot recognize each other, right? So you have to have some specific biochemistry happening in order for a fertilization event to happen. And if that doesn't occur, then you're isolated. So a nice example of this is sea urchins, which basically just release their sperm and eggs into the water, and the sperm and eggs would have to just bump into each other in the ocean. And it turns out if you look at the sperm and eggs from some of these different species, the sperm from one species cannot fertilize eggs from another species. There's just no biochemical interaction there. So again, that the gametes are helping drive that isolation there. Then you've got a few examples here of barriers that are happening after fertilization, and they're either reduced hybrid viability, so you've got the hybrids between these two populations or two different species are just not as viable as the parent species, right? Not as healthy or strong or fast. Or you've got reduced hybrid fertility, right? So this is where the hybrid offspring are sterile. So that's a genetic and evolutionary dead end in that sense. And then you've also got this phenomenon called hybrid breakdown, which for our purposes is basically very similar to really the reduced hybrid viability, where the hybrids are just much smaller, stunted compared to the parent species. So bottom line is, we've got lots of different mechanisms that could help drive reproductive isolation either before or after fertilization. So let's talk for just a minute about what happens if let's say we have two, relatively newly formed species, right? We've had this reproductive isolation. These two populations have diverged into different species, and now those two species come back together and are at least given the opportunity to potentially interbreed. If that's the case, you're going to end up with, maybe a large number of hybrids being produced between these two new species. And so there's a couple of things that could happen, as I alluded to in the first half. If the offspring from if the hybrid offspring of those two species are very healthy and fertile, those two new species may just kind of merge back into one species, right? Because those hybrids are fit, or out competing the parents, and can produce their own offspring. But if the hybrid are less fit, then or are sterile, then you may reinforce that barrier, right? So yeah, we're making these hybrids, but the hybrids aren't as robust as the parents or can't reproduce themselves. So even though we're getting these hybrids made, we're still going to maintain these two separate species. So let me just show you one example where this seems to be the case. And you do have this what's called the hybrid zone. But because of this reinforcement, we seem to be maintaining these two separate species. So this particular hybrid zone, we're in Europe here, and we've got these two species of toads that we think emerged relatively recently. So the yellow-bellied toad that's down here in the yellow region, fire-bellied toad up here, and you can see at the point where their two ranges butt up against each other, we're getting lots of hybrids being produced here in this hybrid zone. Now, in this case, researchers have looked at the hybrids and they see, yeah, the hybrids just don't seem to do as well as either of these parent species on their own, right? They're just not as viable. So even though these hybrids are being made, it looks like we're going to maintain this separation between these two species because the hybrids are not going to outcompete either parental species. So that would be an example of reinforcement in this case. But you could imagine the situation, as I said, if those hybrids were, as fit or even more fit than the parental species, you would just see this hybrid zone growing and spreading over time until eventually our entire map would basically be orange, right? We'd have some sort of blended species there. All right, so last slide, talking about things that could alter the rate of speciation between groups. So, the speed has been observed to vary pretty dramatically depending on what organism we're talking about. The fastest, as I mentioned, would be plants with this polyploidy model. But in general, you're going to see faster speciation in populations that have very specialized diets. So if you have a specialized diet, you're spending a lot of your time in a very specific microenvironment looking for these specific foods. So that can help increase isolation. If you have a lot of sexual selection going on, right? So we talked about that behavioral isolation as one example of that, right? If you're very choosy about what your mate looks like or how they behave, that can increase separation really quickly. And also just if you can't move around very easily, so the more limited your mobility is, essentially the easier it is to get physical geographic separation, right? So, if you're talking about populations of birds that can fly thousands of miles, it's a lot harder to get geographic isolation compared to, say, snails that are moving, very short distances at a very slow rate. So anyway, I hope this has given you some feeling for how we would define a species and how new species could occur through evolutionary change. Thanks for listening.